Hello, and welcome to this webinar titled Introducing Chromassette, a modular chromatography platform enabling next-generation bioprocess purification, hosted by Biopharma Asia magazine and presented by Masayoshi Nagaya, Senior Global Technology Manager at JSR Micro Inc., and Ping Huang, Director, Head of DS Bioprocesses, OED Abbvie at Redwood. My name is Stephen Edwards, and I'll be your moderator. Now, please allow me to introduce our first presenter. Masa Nagaya has more than a decade of experience in CMC operations of bulk drug substance and drug products, product management of membrane and chromatography resin technologies, and process engineering of advanced semiconductor nanolithography materials. Today, he leads JSR's global SSG team in applications and process development for purification and recovery technologies for the biopharmaceutical industry. He received a bachelor's in chemical engineering from Bucknell University and is the author of several technical publications. I will now be handing over to our first presenter. Welcome, Dr. Nagaya.
Thank you very much, Stephen. Good morning, good afternoon to all. My name is Masa Nagaya with JSR Life Sciences, and I'm responsible for the global applications team. Thanks for joining me on this webinar to hear about the technology we're developing called the Chromassette. I'm very happy to be sharing our most recent progress with the development of this Chromassette technology. And following my presentation, we have Dr. Ping Huang from Abbvie present an application of this Chromassette from beta testing the technology. So let me start off by introducing JSR Life Sciences, which is a division of JSR Corporation, a Japanese chemical company. JSR Life Sciences headquarters is located in Sunnyvale, San Francisco Bay Area, and has a global footprint with just over 25 manufacturing and R&D facilities throughout North America, Europe, and Asia. Our Life Sciences division is comprised of two main sectors, Bioprocess, which I am part of, and the other is Diagnostics and Discovery. In our Bioprocess materials business, we focus on purification technologies. And today, we have our protein A resin, AMSTER-A3, in the market for primary antibody capture step. In the recent several years, our Bioprocess family expanded to include KBI Biopharma, an analytical and contract development manufacturing organization, as well as Celexis, a premier cell line development company, enabling really a gene to GMP bulk drug substance development and manufacturing to serve the biopharmaceutical industry. I'd like to start us off with the motivation in current biomanufacturing paradigm. So we have seen growing biosimilar molecules. Just over past few months, we have seen several approvals of biosimilars by regulators, globalized biomanufacturing, so essentially building of manufacturing plants at various local markets. This is, of course, enabled by single-use manufacturing technologies for lower capital expenditure requirements and the ability to really commission a facility very rapidly. We have seen a tremendous improvement in cell culture titers over the years, but that has led to the downstream to be a bottleneck. So there's an opportunity here. And finally, we see new constructs beyond monoclonal antibodies to fragments, bispecifics, FC fusion, and so forth. With these paradigm shifts, we see Chromassette technology to provide a scalable route for higher productivity downstream processes because of its ability to provide resin bed support using a familiar resin chemistry in a reduced footprint and, of course, compatible with single-use technology. So now I'd like to describe to you what the Chromassette technology is. But before I do, I wanted to make a comment that this development of Chromassette platform is a joint effort between JSR and SPF Technologies, which is based out of Boston. So to describe the technology, here I have broken it into three categories, so design, functionality, and capabilities. In terms of the design, the Chromassette, as the name suggests, it's a chromatography in a cassette form. It's rectangular, modular, stackable, and also disposable. What distinguishes the Chromassette device is its unique internal scaffolding that provides supported resin bed. You can see the picture on the upper right-hand side of the PowerPoint. It's an open platform, so we can pack it, and this pre-packed cassette will be delivered to our end users. In terms of functionality, I'll be talking about the details later in this presentation, but it's fully interchangeable with a conventional column format. And because of its unique internal scaffolding, we see an improved pressure flow properties with the packed bed. Finally, for capabilities, it is modular, and you're able to stack the cassette and in a linearly scalable fashion. In addition, the scaling is predictable in terms of the pressure flow because the internal scaffolding is constant for all cassette sizes. It can deliver higher productivity processes because of its ability to run at a faster linear velocity. Finally, this is disposable technology to be used for the lifetime of the resin, so we have a high convenience and quick turnover as a capability. So this is a comparison of what a conventional 100-liter column looks like on the bottom, and what we anticipate a 100-liter Chromassette to look like. We envision a holder with 20 5-liter devices to have a total of 100-liter resin volume. So here is what the actual Chromassette device looks like. On the left, we have what we call our SPD, which is our self-contained scale-down devices. They simply hook up to your FPLC system. The shorter of the two is a 4 mil resin volume device with a bed height of 6 centimeters. The length of the cassette is the bed height. The 20 centimeter bed height device is on the right at 13 mil resin volume. These two scale-down devices will be launched commercially first. We're targeting second half of this year for these SPD devices. The picture on the right is our prototype 1-liter module with 20 centimeter bed height. This is our GMP scale device, and I'll be sharing our 4-stack, so 4-liter volume study in this presentation. The 1-liter device is essentially a 100x scale up of the smaller 20 centimeter SPD. Before we start reviewing the data, I wanted to talk more about the internal lattice structures. As was described before, the cassette has unique internal lattice as shown in this picture. This scaffolding essentially supports the resin beads that are packed. And now here's an image with the device packed with resin. Essentially all the beads benefit from the frictional forces that provide the wall support to the resin. And this wall support allows for the resin to be run at higher velocity, so you no longer get compression as you may see in a conventional column. I wanted to point out that in a column-based system, the wall support is essentially the peripheral of the column as well as the top and the bottom. As you scale up, you start to lose that wall support. So the resin in the bottom center of the column will essentially bear the weight of all the resin above and more compressed as opposed to, let's say, resin at the top. In the Chromassette case, the lattice remains constant regardless of Chromasette's size, enabling predictable scale-up, especially for pressure flow, as well as linear scalability. I'd like to show some examples of how the lattice in Chromassette can alter your pressure flow curve. Here we have a pressure flow study using Fractogel TMAE S resin, so which has an average particle size of about 20 to 40 microns. A small 5 mil conventional column with a bed height of 5 centimeters is shown in blue. And in red we have the Chromassette, which is also a 5 mil resin volume with a bed height of 6 centimeters. And this is plotted in normalized pressure as a function of linear flow rate. You can see that the Chromassette maintains a nice linear pressure flow curve up to about 800 centimeters per hour, while you see the column hit a critical velocity at about 900 centimeters per hour. Even at this scale, you can see the positive impact of having the lattice support. And with this example, we can see that the Chromassette technology enables you the use of small particles resins for Q flow-through applications at membrane-like speeds. Now I wanted to show a scaled-up data, essentially compared to the previous page. Here we are comparing a pilot scale column at 20 to 30 centimeters in diameter which is typically for typical for, let's say, tox engineering runs as well as early clinicals. So we have packed our own AMSTER A3 resin into various formats with 20 centimeter bed heights. So let me walk you through the plotted data sets. We'll start from the bottom. In green, we have a small scale at 1.15 centimeter diameter column. In blue, yellow, and gray, we have the 20 and 30 centimeter column, Axichrome, BPG, quick scale. These are shown under the yellow trend line that is shown in the graph. Finally, we have a single stack 1 liter Chromassette, pressure flow in blue. As can be seen, the 1 liter system closely resembles that of the small scale columns due to the wall support, and linear relationship is maintained. By simply switching from a conventional column to Chromassette, given a limit of, let's say, 2.5 bars, which may be the limit based on your process kit, you can see an immediate advantage of being able to run 70% faster, allowing for a higher productivity when Chromassette is utilized. This technology does not change the diffusional mass transfer. So dynamic binding capacity will change with the flow rate. So to keep the same binding capacity, but to have the faster productivity, you can consider conducting the non-product load steps faster. So that would be process steps like equilibration, wash, and so forth. In the introduction of this technology, I mentioned that the Chromassette technology will be open platform. Here I am showing some examples of various resins we have packed. To date, we have packed over several dozen different resins from various suppliers. Same as conventional column, we'll conduct integrity tests to look at asymmetry, HETP, also hydraulic resistance. So there will be a specification for these parameters, and each device will be tested prior to release. So as we continue our beta test, we continue to expand our resin library. I'd like to switch gears slightly and talk about various chromatographic performances. I'll start off on this slide with a cation exchanger, followed by anion and protein A on later slides. So here we're looking at bind and elute on a cation exchanger. We're using a macrocap SP resin with model feed of lysozyme to look at the breakthrough. And you can see that the breakthrough of both Chromassette as well as the column with a residence time of 4 minutes shows essentially very similar breakthrough curves. Here we have the binding capacity as a function of essentially retention time for the column and Chromassette. You can see an equivalent binding capacity at various residence times. Before each load cycle, we also looked at the carryover, and in both cases, we saw less than 0.1% of the load, suggesting no issues with dead spaces for both formats. Using the same resin in a bind and elute mode, we did a proof of concept to see if we can obtain good separation. We used a cocktail feed of lysozyme, RNase A, and cytochrome C, and conducted a gradient dilution for this 3-protein mixture. On the left is our Chromassette in a 6 centimeter bed height, and on the right is a conventional column at 6.2 centimeters in bed height. We can see that the resolution is similar for both, with peak centers that occur in the same position in the elution gradient. This was a good proof of concept to see that the separation capability of the resin is maintained in Chromassette. Our partner KBI looked at utility of Chromassette for flow-through intermediate polish steps, as AEX is widely used for processing MABs and MAB-like molecules. They had included the Chromassette cassette as part of their screening, as they were interested in looking at how the Chromassette compares versus conventional column for selected TMAE, S, and also M resins. What they found was that the yields were high, as well as the Chromassette format did not alter the selectivity relative to the high molecular weight component, as well as the host cell protein removal from the feed. Where they observed the difference was the ability to run with favorable pressure flow characteristics, which meant that the ability to achieve higher productivity at shorter resonance times, particularly for higher concentration streams. From this initial beta test, they favored this module over membrane chromatography since they were able to utilize resin that have historical knowledge about and familiar chemistry to effectively remove the process impurities. So the next few slides will be on the performance of for capture step. We're using our AMSTER A3 resin for these studies. Using a scale-down device, we looked at the effect of modularity by stacking the cassettes. In this study, we assembled 4 stacks of 6 centimeter bed height, as well as the 20 centimeter bed height. The device looks slightly different from the SPDs shown before, as they require a holder, which can be seen on the right. When stacked, the feed will be distributed evenly through the top and permeates through the bed in parallel across the 4 units to keep the same bed height. In this study, we looked at the effect of pressure drop as a function of linear flow rate when each of the 4 devices are separately, as well as when they're stacked. In green is the 6 centimeter bed height cassette, tested individually as well as stacked. And in blue is the 20 centimeter bed height of the individual and the 4 in assembly. The pressure flow will be lower for lower bed height, as shown. And we can see here is that the 4 individual cassettes, regardless of size, we see less than 10% variation for the pressure flow between the cassettes. In addition, the 4 stack assembly is essentially the average of the individual cassettes. To further study the stackability, we also ran a protein polyclonal through the column to look at resin performance. On the left, we have a bar graph of the binding capacities at 2 resonance times, and on the right, the respective elution volumes. Similar to the previous slide, we looked at individual cassette performance, which is shown in the solid colors, and also the performance when the 4 are assembled in a stack, which is represented as pattern bar here. We can see that both the DBC as well as the elution volumes of the 4 stack matches closely to when each of these cassettes are tested individually. With this data, we can see that scaling through stacking really does not alter the performance. Now, I'd like to switch gears and talk about scaling up for GMP use. This is the most recent data we have collected, and I'm very excited to be speaking about the scale-up proof of concept that we conducted. So here on the picture, we had the 1 liter device you saw earlier, and the bottom is the 1 liter device in a prototype holder so that it can be connected to a skid. Now we have a 4 stack of the 1 liter module with 20 centimeter bed height, making this stack a 4 liter equivalent resin volume. The resin packed inside is our own AMSTER A3 resin. The very right-hand picture shows the size of a 4 liter stacked module when it is connected to an active process in our applications research lab in North Carolina. The holder is acrylic and is really just for prototyping. In parallel, we are working on a commercial stainless steel holder with a disposable flow path. So with the 4 by 1 liter stacked, we looked at pressure flow performance. We have a few data sets here, so let me walk you through the legend. Again, all these data are packed with our Protein A resin. So in brown/red color, we have a reference scale down with 20 centimeter bed height with resin bed volume of 13 mils. In navy blue, we have a pilot scale, 20 centimeter diameter oxychrome. The line is actually shown on the left-hand side of the graph with the higher pressures. In green, light blue, yellow, and gray, we have the 1 liter Chromassette devices tested individually. As we have seen before, the lines are all linear to 1200 centimeters an hour. In orange and blue at the top of the legend, we now have the 4 stack of the 1 liter devices assembled in different orders. So you can see that the pressure flow property of the 1 liter Chromassette is very similar to that of the scale down SPD device, and minimal lot-to-lot difference between the devices. In addition, the pressure of the 4 stacked was comparable to that of the single cassettes. We are actually very encouraged to see such similar scalability performance between the 13 mil and the 4 liter, which is about 400 times bigger in size. So, and again, we can take advantage of the faster flow at scale when using the Chromassette compared to a conventional column. So with these 1 liter GMP cassettes, we also ran purified polyclonal to look at its binding capacities. On the top, we have the DVCs at 10% breakthrough of various devices and columns. So let's focus on just the orange bars, which is the binding capacity at 2-minute resonance times. So we tested this polyclonal on 3 stack of the 1 liter, 4 stack of the 1 liter. Each of the modules ran separately, so each of the Chromasets ran separately. A scale down of the SPDs at 13 mil, and also a reference scale down column, 0.5 centimeter in diameter. In all cases, we're seeing a DVC of about 46 to 48 mg per mil. On the bottom graph, we have the summary of this at various retention times. So you can see that the DVCs overlay very nicely. With this data, we can see robust scalability from lab scale to pilot in predicting performances, as well as the stackability of the modules at scale. We also observed minimal lot-to-lot variation in DVC from the various cassettes. So this is the last section of my talk. Before I pass on to Dr. Huang, I wanted to share with you on what the possibilities this technology really enables. Resin suppliers like ourselves, when we design resins, there are various factors and trade-offs we must consider. And one is mechanical stability, so that it can be implemented for essentially large-scale process chromatography. With the lattice support, we can move away from focusing on mechanical properties of the resin, but to really focus on things like mass transfer, binding capacity, and selectivity. For instance, with today's resins, you cannot have to open up a pore structures due to fragility concerns of the bead. So as an example, we started to explore what we call hyperproductive processing through the use of 30 micron resin we developed for this application. The 30 micron bead is too small to be used for a conventional column. However, it can be implemented in a Chromassette. We conjugated our A3 ligand onto the 30 micron and compared the performance versus A3, which is a 50 micron product. You can see our answer A3 here in the curve. At the lower retention times, you can see an improvement in DVC by about 70% by simply going to a smaller bead size. We can start to think about processing at these lower retention times to take advantage of this phenomenon. So with this prototype 30 micron A3 bead, we did a 10-cycle proof of concept using a 2-column system. We explored resonance times of between 0.6 minutes to 1 minute, and where the cycle duration was essentially about 15 minutes. So this is a very quick cycle. With this process, we were amazed to see an incredibly high productivity. Just as a reference, today's published productivities are about 15 to 25 grams per liters per hour. And with the use of this tailored resin in hyperproductive mode, we were able to achieve 198 grams per liters per hour with low buffer consumption as well. Here we have an overlay of the 10 elution peaks to show that this process does not cause peak broadening, so we will not see amplified dilution of your drug substance. With this hyperproductive process, we also looked at impurity clearance. In yellow, we have HCP, while in blue, you have DNA. Looks like we can't see the legend here, but In both HCP and DNA clearance, we observed about 2 log or greater removal, and the recovery stayed above essentially 98% for each of the cycles. So scaling up this hyperproductive process, we did some basic calculation on what this could mean. Essentially, 200 grams per liters per hour productivity is achievable using a regular skid if the titers are greater than 9 grams per liter. Using an active process skid with 3 5-liter Chromassette cassettes, you can capture about 3 kgs of mass per hour. Now, if we had a 2,000-liter bioreactor with a 10-gram titer, the capture step can be completed under 7 hours, which is an incredible amount of product in a short period. So now I'd like to summarize my talk. The Chromassette is a high-performance stackable cassette device that can be packed with any chromatography resin. And its unique support that offers the ability of a performance of a column, but with improved pressure flow, which are not altered by scaling. It is disposable and pre-packed, so you can have a quick turnover. And finally, the Chromassette can enable next-generation bioprocessing, which Dr. Peng will talk about more next. So please stay tuned for a commercial launch of our scaled-down SPD devices in 2019. And the GMP scale devices will follow in 2020. So I wanted to recognize many people involved in the development of this technology from SPS, as well as my colleagues at JSR. It really does take a village to launch a new technology like this, and I'd like to recognize our partner company and early tester KBI, as well as the Abbvie team for providing data and feedback on this technology. And thank you very much for listening to me. I believe we're taking questions later, so I'd like to give the stage to Dr. Peng Huang. Thank you.
Thank you, Dr. Nagaya. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now please allow me to introduce our second speaker. Peng is Head of Drug Substance Biologic Processes for Oncology Early Development in Abbvie. He obtained a PhD in Biochemical Engineering from the University of Maryland, Baltimore County in 1992. After postdoc in NC State, he joined Protein Design Lab, which was later acquired by Abbvie. In the last 23 years, he led teams of scientists and engineers in bioprocess dev, tech transfer, and scale-up. He worked on projects in at least 30 INDs. 2 of them successfully progressed to BLA and FDA approvals in 2015 and 2016. I will now be handing over to our second presenter. Welcome, Dr. Huang.
Thank you for the introduction. Can you hear me okay?
Coming through nicely.
Okay, good, great, thank you. So I'm going to present from the end user's point of view in a biopharmaceutical company. I want to thank JSR for the introduction and the collaboration, and also thank many other companies such as GE, Millipore, or Sartorius, or other companies for developing many cool processing technologies for us to use so we can produce and purify pharmaceutical products quickly and safely. Of course, we don't only say thank you, we pay a lot of money for the technologies too, such as paying $20,000 per liter of protein A resin, or $1 million for an Amber system. So, joking aside, I want to give the brief introduction of the bioprocess history. When I was in the graduate school in the Department of Biochemical Engineering in the 1980s, many techniques were being developed for the production of therapeutic proteins, including at that time a very promising transgenic animal production technology. However, in the 1990s, the mammalian cell culture technology became the dominating technology and adopted in our industry. During that time, I was able to contribute a little to the formation of current purification platform process in the industry. By the end of the 1990s, the current platform process was established across our industry using CHO cells or NS0 cells for production and protein chromatography for purification. To be more specific, next slide, please. So I do the forwarding. Okay. Our current bioprocess starts with a cell bank vial and expands to production bioreactor in about 4 to 5 weeks. The bioreactor is harvested by centrifugation or by depth filtration, followed by protein chromatography and low pH virus inactivation. The fine purification involves the anion change and other polishing chromatography, followed by nanofiltration, ultrafiltration, and formulation at the end. Current platform process has been successfully scaled up to 25,000 liter scale and used to produce many blockbuster drugs. Over the last 20 years, many improvements have been made, mainly in the use of disposable components in the process. However, there is no major change in the unit's operation. The current process is also very complex. It is operated in a batch mode. It is difficult to automate. A larger scale manufacturing facility takes 4 years and billions of dollars to build. So it is very slow to respond to an increase of demand, or a huge investment has to be made before a drug is approved. It's a risky business. So I think our current platform process has a lot of room for improvement. Two years ago, one of the scientists, Bob Duffy, in our bioprocess development group, was curious about the Chromassette and started testing in our lab. I was very interested on it too, because I know the column chromatography is not robust, and it is difficult to operate because if the air gets into the column, a crack will be created, and the whole batch of several $1 million will be ruined. So the chromatography are always performed with a sophisticated liquid handle resistance. However, with Chromassette, column is converted into cassette. Masa just talked about it. And it's easier to scale up just by simply put several pieces together, just like putting several UF membranes together. Also, the feature I like the most, Masa also mentioned that the packed resin is supported by scaffold, so the resin cannot move around, even one is compressed, or in some extreme case, if air is accidentally introduced. So to test this hypothesis, we introduced the air purposely into the Chromassette and measured the HETP before and after air intrusion. Results showed that the HETP was not changed, and the symmetry was slightly better even after air intrusion. So this demonstrates that it is increasing the robustness by using the Chromassette technology. We also performed some head-to-head comparison of plotting a column versus a Chromassette chromatography and found minimum differences. The dynamic binding capacity was almost identical at the 68 mg per ml. Step recovery was identical at the 93%. The protein area was a slightly lower concentration for the Chromassette, but the other impurity clearance were comparable. So we think it is very promising technology, and Masa also has shown a lot of other tests as well. So I think in the near future, if the Chromassette can be scaled up successfully, it may be possible or easy thing to replace most of the columns with the Chromassette without too much change in the overall process. However, I think this is only a marginal improvement. What I'm really excited about is the geometry transformation from column to cassette format. With this transformation, the whole process on the right-hand side and the fine purification, from the harvest to formulation, is composed of only membrane filtrations and cassette operations. So I proposed a drastic change last year at about this time in a meeting in Boston, that these different unit operations in the harvest and purification should be all integrated into one format and operate as one unit operation. So this is the concept of the integrated bioprocess. On the left-hand side, the single-use bioreactor will replace the stainless steel one because of high titer and perfusion cell culture processes we have been seeing in or in the conferences or in the report. On the right-hand side, the harvest and purification is integrated into one unit operation. I borrowed this picture from Sartoria to demonstrate that in this integrated recovery purification trend, the disposable components can be replaced easily. So this IRP trend will start with a couple depth filters to remove cells, followed by plotting a Chromassette, or GE has been working on promising plotting a filter. And then the viral inactivation chamber, and then followed by another depth filter, a Q filter or Chromassette, and another Chromassette for polishing and virus filtration. The purified bulk can be filtered into a point-to-micron filter into storage bag. So one IRP trend can be performed in a batch mode. But 2 to 3 IRP trends can be used in a continuous mode to process harvest continuously. I have demonstrated the proof of concept at a small scale that the batch harvest and purification could be completed in 6 hours. We are working with JSR on scale that up to pilot scale with a 1 liter plotting a cassette, and to hopefully to the GMP manufacturing scale in the near future. I think although Masa said it's possible to scale up to 100 liter, I think with the IRP trend, a 10-liter protein A Chromassette may be a good scale for the industry application. So I have presented this concept of integrated recovery and purification, and also the experimental results in the couple of conferences in Boston and in California last year and this year. And I will be presenting again in August in Boston on the Bioprocessing Summit on continuous processing with some new results. Hopefully we can have some pilot scale results to demonstrate that we can purify 50 grams of the antibody in just 6 hours. So if you are interested, please come to the meeting. Ultimately, I hope my concept of the IRP trend can be adopted within our biopharmaceutical industry, similar to the adoption of the integrated circuitry in the electronic industry. If this process trend or process chip can be widely adopted, the connections between the different unit operations in downstream will be standardized. And the IRP will eliminate most of the process intermediate tanks or bags. It will significantly reduce buffer usage and the number of buffer tanks. And more importantly, it will drastically reduce the manufacturing facility construction costs and the timeline. So the key reason that the facility construction costs will be significantly reduced is because in the later stage or commercial, there will be no scale up or scale out. At the early stage, 1 or 2 IRP trend can be used to produce FIH early stage material, but in later stage, the same scale I envision the 10-liter protein A trend would be sufficient to produce later stage and commercial scale up to 1 ton of the antibody per year. Just run these 2 to 3 trends continuously, days and night. And because the process is robust with the Chromassette, and the operation of replacing the disposable components are very easy, we can use the robots to do the job. So the operators will not be on the manufacturing floor, but will be sitting in the control room, comfortably monitoring the operation. This will eliminate the chance of contamination introduced by human operators. And also because there is no scale up, these trends can be housed in a very small area, so the environment can be kept very clean to prevent the contamination. So this ultimately will benefit our patients by reducing the manufacturing cost and increasing the safety of our product. So overall, I'm very excited about the Chromassette technology that has the potential to be used to integrate harvest and purification into one process trend. And this will drastically reduce the manufacturing footprint and construction cost. Together with the perfusion and high-titer cell culture processes, a completely automated continuous production and purification of biopharmaceutical products may not be too far in the future, and this is my vision, and thank you for listening.
Thank you, Dr. Huang. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now moving onto our first question, we have, does the Chromassette always come pre-packed, or can it be packed by the end users? I believe that's for you, Dr. Nagaya.
Yes, thanks, Stephen. The Chromassette requires a unique packing station, so we will only be providing end users with pre-packed columns. We also think that this allows the end users to not worry about the headaches that come along with packing various types of resin that essentially have unique packing requirements.
Thank you. Our next question is, when do you expect to launch the 30-micron resin? Again, for you, Dr. Nagaya.
Yes. We have not identified a product launch date for the 30-micron resin. We're still investigating the utility of the resin and its applications through essentially beta testing. So if anyone is interested, please get in contact with me, and I'd be happy to speak about more about this.
Thank you. Now, unfortunately, I think that's all the time we have for questions. But I would like to thank our audience members for your questions. And before I finish the webinar, I'd like to ask our presenters, Dr. Nagaya and Dr. Huang, if they have any closing remarks. So, Dr. Nagaya, do you have any closing remarks?
I'd like to thank everyone for your time. Please feel free to reach out to me with any questions. And I hope this webinar on our Chromassette technology was interesting to you all. Thank you.
And Dr. Huang, do you have any closing remarks for our audience?
I just also thank you for organizing this webinar, and hopefully we can reach to more people, and our industry can move into a more efficient production of the biopharmaceutical product. Thank you.
Thank you. And I would like to take this opportunity to thank our presenters. Once again, that was Masayoshi Nagaya, Senior Global Technology Manager at JSR Micro Inc., and Ping Huang, Director and Head of DS Bioprocesses OED at Abbvie at Redwood, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers to you. Thank you, everyone. Have a great day.